Hi, I'm Dr. Natalie Thiex, and this is my laboratory. The purpose of our lab is to research macrophage cell biology. Macrophages are white blood cells that you might have heard of before. They're part of the immune system, and their job is to eat up the pathogens like bacteria or viruses that come into our bodies. And they also even eat up infected cells or even cancerous cells. And the point of our lab is to understand fundamentally how do these cells work? How do they do the processes that they do that allow them to fulfill their function in the body? And so we take a variety of approaches, or research approaches, to answering that question. But one of the things that we have done is to use a CRISPR-Cas9 whole genome screen to identify important genes for regulating processes in macrophages. So some of the things that we've studied so far are genes that regulate the process of macropinocytosis, which is cell drinking. And we've also done screens to identify genes that regulate lipid droplet biogenesis and lipid droplet metabolism. And the way that these whole genome screens work is we are able to get a little tiny tube of RNA that has guide RNAs that are complementary to all of the genes in the mouse genome. And these guide RNAs will target an enzyme to the DNA, and it makes a double-stranded break in the DNA, which basically disrupts the gene function because it makes a frame shift mutation and makes it so that gene cannot make a functional protein. And so by taking all of these different guides and putting them on millions and millions of different macrophages in a dish, we're able to knock out basically one different gene in each cell, and then ask, does that cell with that gene disruption have the ability to do the process like macropinocytosis? And so we have this really essentially a huge experiment that we're doing all at once, which is kind of the idea of systems biology, that we're able to investigate how the disruption of one gene has an impact on the whole cell. So using the guide library tool, we knocked out all of the genes in the mouse genome one at a time in different cells. And then we asked ourselves, how does this gene disruption, using the new technology CRISPR-Cas9, how does that have an effect on the function of the cell? And because macropinocytosis is cell drinking, and I always had a student who used to use a big gulp, a 7-Up big gulp, to demonstrate the idea that these cells are basically rearranging their membrane and gulping in large volumes of the medium around them. So if you make the medium around them fluorescent, you can tell if they're doing this process because they have big fluorescent vesicles inside of them. So using a flow cytometer, which can measure the total fluorescence of a cell, we asked which ones are bad drinkers after the knockout and which ones are good drinkers. And sometimes we call it low drinkers and high drinkers. But basically the gene disruption, if it's a gene that's important for macropinocytosis, if we knock it out, then they won't be able to do macropinocytosis anymore, and they won't be able to take up this fluorescent medium. So we use a flow cytometer to sort those cells into different tubes, and then we actually use DNA sequencing to sequence the DNA of the cells in the tube that aren't able to do macropinocytosis. And from there, we can use our bioinformatics tools to figure out which genes are important for the process of macropinocytosis. So that's my little spiel. Should we just introduce ourselves quick, and then so we can maybe go around, everyone can say their name, maybe where they're from, what year they are, and then after we do that, then we can just go one at a time and everyone can say what their project is. Yeah. And then if I always have a million. I like to talk, so I'll probably add more than is necessary. All right.
Well, my name is Tate Buckled. I'm a senior undergraduate here in Dr. Thiex Research Lab, and I am from Hartford, South Dakota.
Hello, my name is Lu Huang. I'm from China, and I finished my PhD in SDSU, and now I'm working as a postdoc in Dr. Thiex lab, and this is my second year of my postdoc study.
My name is Jared. I am also from Hartford, and this is my first year as a graduate student.
I'm Nick Steinmeier. I'm from Sioux Falls. I'm a junior undergraduate student.
I'm Louise Lunga. I'm from Congo, and I finished my master's here in the summer, and now I'm working as a research associate.
All right, so you just want to say what you're doing?
So from the whole genome screening that Dr. Takes talked about, I basically just am identifying some of the top hit markers, and I work on specifically something called the Manus receptor, and I knocked it out, and then I compare the phenotype of it when to the control cells while it's undergoing macropinocytosis, and just identifying what looks the same and what looks different.
So for me, I worked partially also with what Dr. Thiex just described, but also I'm working also in transcriptome study. Basically, you can look at the RNA level of the cell of the macrophages, and by that you can know the protein expression level in the macrophages, so that you can understand the protein functions in the macrophages. And so that is also actually very helpful for understanding the big project about the macropinocytosis, the mechanism in macrophages.
I too also worked on the Cas9 screening for macropinocytosis, and I spent a lot of time learning how to use computers to actually analyze the high and low linker sequencing data so that we could determine which genes were actually relevant to each population, so that we could analyze to see which genes would actually affect the process the most.
I will just add a few things since I'm thinking them at the time. But I do think it's important for prospective students in your new undergrad or graduate students to consider the possibility of going into a systems level biology project or using bioinformatics or coding or applying math to the study of biology. It's not as hard as you think. It's just a series, it's just learning a set of skills, just like learning anything in the lab. But it's a very powerful tool to have because you can make things so much faster. It is so completely adaptable. Pretty much your imagination and the sky is the limit to what you can do, what kind of problems you can solve, what kind of data you can analyze using coding. And it's also a really hot job skill to have. If you can handle data with a computer, if you can apply math to biology, if you can analyze a big data set, then I think you can get a job really easily in a variety of different fields afterward.
Okay, so what I'm working on is looking at genes that help regulate actin formation and whether or not they play a role in the formation of macropinosomes, and also to confirm or deny if the screen data was correct or not.
I'm also working on the same project, looking at genes that are important for macropinocytosis and actin regulation. And when we knock out the genes in the macrophages and observe the cells and try to understand their biology.
Well, I can just sort of go back and summarize some of the different aspects of our research that would be considered systems biology. So we've used CRISPR-Cas9 whole genome screens where we were able to use different guide RNAs and the newly discovered endonuclease Cas9 to knock out all of the different genes in the mouse genome in different cells in a large pool, and then from there be able to assess the phenotype of those genetically disrupted cells to identify genes that are important for a variety of macrophage cellular processes like macropinocytosis, lipid droplet biogenesis, and lipid droplet catabolism. In addition to that, we've also done transcriptomic experiments where we used RNA-seq, which is basically using a DNA sequencer to sequence all of the different RNA in the cell. So that gives us a picture of what genes the cell is expressing. And then from there, we're able to see how different exposures or gene knockouts affect the transcriptome or affect the gene expression in that cell type, which then may affect its phenotype. What else? What's that? Oh, and then we also use high content microscopy in order to be able to screen a bunch of different gene knockout phenotypes at one time. And oftentimes we analyze our fluorescence microscopy data using different software that are able to recognize and quantify the fluorescence on a cell-by-cell basis. And in addition to that, we even have a component of that software that has a machine learning aspect to it so that we're able to have the computer itself identify different cellular phenotypes and quantify them. We've summarized our entire research in 15 minutes.